Well, welcome everyone. I think we'll get started. My name is Vera Trainer. I am the Marine Science Program Director here at ONRC as of last January. And there are some folks who've been here this week to work on writing a paper on harmful algal blooms. So we're very, very lucky tonight to have Dr. Donald Anderson, who's from the Woods Hole Oceanographic Institution. Don has been involved in HAB research, harmful algal bloom, or red tide research, for several decades. I think I was trying to calculate how long I've known him, and it's over 30 years. He's done some really interesting work in the Gulf of Maine on some of the red tides there. And then he's taken his work really around the world to provide training to many folks who are interested in this field of research. So more recently, he's been involved in work in Alaska to determine some threats there to the indigenous populations and to the marine wildlife there, which is what he'll be talking about today. And I think many of us in the field see Don as sort of the godfather of harmful algal blooms, because he's really led the way. He's testified before Congress, many times, and he's really advocated for this work. And I think I have my career to thank Don for, because without him, a lot of the funding, a lot of the government support for our work would not be there. So tonight he's going to be talking about toxic red tides, an emerging climate-driven threat to subsistence communities and ecosystems in the Pacific Arctic. Thank you, Don.
All right. Well, thank you, Vera. And thanks to all of you for showing up fairly late at night, and you on Zoom as well. Thanks to my colleagues who are here at the same meeting for joining, and you can see they're all sitting in the back of the room because they've been drinking wine, and they think I won't notice if they fall asleep. But I can see you guys. So anyway, this I hope will be a really interesting and a worrisome, I'm afraid, story. A story that is about climate, but it's also about waters not too far from here, and some things that are going on, and a true challenge to the communities there, but a challenge to us as scientists as well, as to how we can help manage and how we can help deal with these problems. Trying to change, there we go, that'll do it. First, can we do the lights, Vera? We get the lights. I never have time at the end to thank everybody, and there's this what I'm talking about is a huge team effort. You can see all these people here from my lab, from other labs, funding sources down the bottom. This has been both a fascinating and just a really challenging project over the last 5 or 6 years that I'm trying to summarize today. So to start off, make sure you're all on the same page with us. If we took some of the water from out here, put it under the microscope, concentrated it, this is what we might see. These are all little single-celled organisms for the most part. Many of them are algae, single-celled microscopic algae. These little golden ones in particular are toxic. Thousands of species in the ocean are there to give us the oxygen we breathe, and they're the base of the food chain, but among those are a few hundred that either make toxins or cause harm in other ways. And this guy here is called Alexandrium catenella, and it is a very toxic organism. Some of you might be old enough to remember Francis Gary Powers, remember the U-2 pilot shot down over Russia? Well, the old, he was supposed to be having a poison pill. He actually had a silver dollar that was coated with the toxin from that organism that he was supposed to have bid onto, like for good luck or something. It was supposed to kill him. He never did. And they had to exchange for him. But anyway, that was the toxin produced by this organism. And it and a number of other species produced this is just a subset of the problems we call harmful algal blooms. These are the ones where there's a poisoning syndrome, a human poisoning syndrome from eating shellfish that have in turn eaten different species of algae, and those toxins get accumulated in the shellfish and can make you very sick or kill you. And you can see we have these acronyms, PSP, NSP, and so forth, and 4 of those 5 are problems for you here in Washington. The one you don't have is this NSP, the neurotoxic shellfish poisoning, which is pretty well confined to Florida. But the one I'm really focusing on is PSP, or paralytic shellfish poisoning. And just a few mussels, a few clams that have concentrated this toxin can not only make you very sick, it can kill humans and many animals as well. And this is the problem that is now moving up into the Arctic. So here's some more background. This is the organism, Alexandrium catenella. See 2 cells here. It's basically about barely visible to the naked eye, size of the sharpest point of a pin. This also is Alexandrium catenella though. It's seed, it's resting state. It's like a seed for a tomato plant or something that most of the year, 8, 9, 10 months of the year, the organism is in this state, in the mud, in a dormant state. And as you see over here, at some point, and we'll talk a lot about why, this cyst, as we call it, a cyst, will germinate. It'll go through meiosis, and the cells divide, and you get 2 cells, and then you get 4, and then you get this bloom, as we call it, of vegetative cells. These cells that are photosynthetic and swimming and making toxin, getting eaten. Then at some point, that bloom forms gametes, little small cells that fuse together to make these really large swimming zygotes that then become that cyst. So that's important. You'll see why we care about those cysts. Now, one of the things my lab does is keep track of a lot of HAB events all over the world, and this is a map we produce for PSP events, or PSP detectable toxin levels of PSP. And we've built it up over the years, and it's got a lot of fascinating things about it, but my eye, these days in particular, tends to look up here. Look at all along above Asia. Look at all above Canada, and over here, and above Alaska. What's the story there? Is this organism? And there's 2 or 3 species that cause this, but many of them, or many of these red dots, are caused by this Alexandrium catenella. Is it not there? Is it not there in abundance enough, or is it we're just not measuring it? We don't know. So that was one of the questions I had, and it got all the more pertinent. As many of you may have heard, the Arctic is warming 4 times faster than the global average, right? Now, why is that? It's real easy. You take an ice-covered landscape, shine sunlight on it, you can see 90% of that sunlight energy is reflected, 10% might be absorbed as heat. If that ice melts, the opposite happens. 94% of that energy goes into the water, 6% is reflected, and you have this huge difference as the ice melts of how fast the waters warm. And that's what's happening in the Arctic, because the ice is definitely retreating. So the first thing I did years ago, I got some funding and joined some German colleagues on one of their research boats. You can see it there, the Marion. And we were on 2 cruises. One, and that you see on the left, along the west coast of Greenland. And this one, in 2017, on the west coast again, but starting a little further up and going a little further north. And it was all to try to look, in my case, to look for Alexandrium. The idea was this is really a cold area, no real history of this PSP problem there. If it's going to warm up and this organism is going to be there, we'll set a baseline. We'll see it's not there now, but it's there 20 years from now. Well, we know something's changed. But all right, let's try to look for it. Let's look for its cells. Let's look for its cysts. And I'm going to compress a whole lot of work in just 2 slides. This is just a little stretch of the west coast of Greenland. You can see this fjord here. The size of these circles tells us how many cysts we were seeing in the sediments. And you can see they're all small. These numbers are 1 to 20, 20 to 40. It's only when we get over to Iceland do we start to see bigger circles, 200 to 400. So they're there, much lower though than we've seen in other parts of the world. And if I were to summarize everything we saw on those 2 cruises in Greenland, we saw only a few swimming cells in all of the waters we took along those. That was really 2 month-long cruises. So the cells were there a little bit. This is August when they should be growing the best. They were there, at very low abundance. When we look at the cysts, we found cysts at virtually every station we looked at. Every mud sample brought up, we could find cysts, but the numbers were really low. 10, 20, 100 cysts per cubic centimeter. That's just very, believe me, a very low number. And of course, one of the explanations is the water was really cold. The bottom temperatures were around 0, maybe 1 degree, 2 degree. Surface water is a little warmer, but where the cysts had to germinate from were really cold. And so the conclusion simply was the Greenland coast is just too cold to support large blooms of Alexandrium catenella. But there are low abundance of cells, so it's there. We've established a baseline against we can assess future observations. But it was kind of disappointing. You know, you go do all this work and you find almost nothing there. It's valuable scientific information, but you know, I wanted more. And by going off to the west, we got a lot more. So moved from Greenland over, got some funding to start looking over here in the northern Bering Sea, up through Bering Strait into the Chukchi Sea of Alaska. And part of the reason I was able to get funding is one of our colleagues wrote this great paper in 2016 where she looked at 13 different species of either stranded or harvested marine animals. So all these different species with different food sources, different habitats. And you can see there's two colors there from all these animals. The red is one of those poisoning syndromes I didn't really mention. It's called amnesic shellfish poisoning, or ASP. And it's up there, you can see it. These animals all have some of that in their tissues, but not as much of a worry to us as saxitoxin. That's the PSP toxin. So what you're seeing is that we've known for a long time. I mean, Washington's way down there. You know you have a PSP problem here. It extends up past Vancouver, all along the Gulf of Alaska, as a real serious problem with PSP. It's been known for a long, long time. So everything down in that lower right, it sort of makes sense that things would show up with these toxins. But when we get up here, there's really very few records of people getting sick or dying. And yet these animals, walrus, whales, are all showing both saxitoxin and amoic acid. So the toxins are there. Interesting. This doesn't say how much of it's there, but they're there, and that's the kind of thing sometimes you need to help you get funding. The agencies say, well, yeah, maybe there's a problem. Maybe we'll give you some support to go look. So we were able to get some support, and many times we've run many cruises where other people had the boat and we were just passengers on it collecting samples, and sometimes we had our own boat, and you'll see about that, dedicated to our own sampling. Typically it'll be a boat like this. This is the Healy, an icebreaker that the Coast Guard operates. We would take both simple water samples as well as cores, taking bottom samples of the mud. And you see this map here that is a typical cruise where might start from Nome and go over here to St. Lawrence Island, and each one of these little lines is a transect where you take a lot of samples and then you move to the next one and have a transect, and then you go all the way up and then eventually come back. That's the kind of data that we would collect. And the other bit of background to remember here are these currents. You can see down in the Bering Sea, there's one arrow on the right that is the Alaskan coastal current. Comes up here from the Aleutians. On the left is this Anadyr current. Goes through the Russian side, the Gulf of Anadyr. And then this water just jets through Bering Strait. And it goes in different directions, including the Alaskan coastal current. Runs along the coast here. You'll hear me talk about Ledyard Bay. That's right there. Kotzebue Sound is right here. Point Barrow, or Utqiagvik now, is right here, the northernmost point of Alaska. But you also notice this whole area is called the Chukchi Sea. So that's just to get your bearings of what we're going to talk about. So, right to the data. What we did right away, if you're studying Alexandrium, you've got to study its cysts. Where are they? So we mapped out the cysts of Alexandrium catenella. What you're seeing here on the right are data from 3 cruises averaged together. And what it shows you is the spatial extent in the general abundance of the cysts of this organism. And when I first saw this, I was honestly astounded. And I'll tell you why, for several reasons. The first is these numbers, this red area here in the middle and another one over here by Barrow. These are among the highest. There's only one other record anywhere in the world with a higher concentration of Alexandrium cysts, and that's just over nearby in Russia. Higher than anywhere else in the world, by a factor of 10, really. Excuse me. So there's a lot of cysts there. Also, this area is huge. What we call a cyst bed or a seed bed is bigger than any other feature we've seen anywhere in the world. And so we picked up two of these sort of accumulation zones or cyst beds, one here and one there, with a little less in between. Very little down here you see in the northern part of the Bering Sea and Bering Strait, and nothing over here in the Beaufort Sea. So, but huge, huge numbers. And just this last year, 2022, we thought we'd sort of seen it all. We got an opportunity to get more samples down here from the eastern Bering Sea, and now we've added that into that cyst map. And you can see yet more of these red bullseyes. So there's 3 or 4 of these big accumulation zones with incredibly high cyst abundance. And this area, if you look at it all, well, the best way to look at it, see this little tiny little inset over here? That's the Gulf of Maine. That's Nova Scotia to Boston, New England. That's where I've been working for years. They have annually recurrent outbreaks of Alexandrium closures with toxic shellfish. And look, that's drawn at the same scale in abundance and in size and area as everything else. You can see your seedbed out here in the Pacific Arctic totally dwarfs anything on the East Coast, anything anywhere else in the world. It's incredibly dense, incredibly worrisome. And we just added this southern leg to it in the last year or so. So it says here, taken together, this is without doubt the largest and the densest accumulation of Alexandrium cysts ever observed globally. And the abundances in the area are truly enormous. And notice that we're not allowed on the other side of this treaty line. And a lot of our high abundances go right up to that line. We can't go into Russian waters. So it's even bigger than we can show here because we can't sample in Russian waters. And you look down here, these are the numbers. The area of it is basically 10 times bigger than the Gulf of Maine. The concentration of cysts we see there is 15 times denser. So there's a lot of cysts. So what's the story? Why is it big? Are they functioning? That's really, are these cyst beds actually doing what they do in the Gulf of Maine, where they will germinate in right about now, March, April, May, make a bloom, forms new cysts, drops down, they're dormant all winter, and they do it again the next year. Is that happening up there? Well, we think, in fact, we know that it's happened in 2018 and 2019. And part of the reason we know is this curve, which I'll explain. Don't worry, it's really quite simple. What this is, we took cysts from all these different places, the Gulf of Maine, Cape Cod, the Chukchi Sea, and we incubated them at different temperatures. And just how long does it take for them to hatch, to germinate? 5 days, 10 days, 20 days, and that's what's plotted on this y-axis. And you can see that all of these different places, all these different cysts follow that beautiful red line there. That's not a mathematical fit to the data. What that line is an 85-degree day curve. You may have heard of degree days, if you want to know how cold was the winter. It's a metric that does both time and temperature. Horticulturists, they use when is your tree going to flower, when are insects going to come out. So it's a way of keeping track of how long you experience a certain temperature. And the way to understand it is that's 85-degree days. So if I incubate cysts at 10 degrees, they take 8.5 days to germinate. What happens if I come way over here and do it at 1 degree? What's that? 85 days to germinate. That's 85 degree days. So why do I go through this? Well, these were the bottom temperatures there in the Chukchi Sea in 2018 and 2019. You can see they were 7 to 9 degrees Celsius. Very similar to the temperatures we've seen in the Gulf of Maine, where this phenomenon occurs every year. So, no question, that organism can function, can germinate, those cyst beds can work. And how much can they matter? Well, here's another set of data. Here we've taken those two seed beds, one Ledyard Bay, one Barrow, and you just look at the temperature binned into 5-year intervals. And you can see that from 1999 or so, that the temperature in the bottom waters has gone up almost, you can see, from 4 to 6 or 7 degrees. And down below, we can actually use that function I just showed you about germination to actually predict how many more cysts are we going to get germinating in a bloom season because it's warmer by that much. And the answer comes down to be roughly not quite a factor of 2. So some people think, well, factor 2 is big. Well, my colleagues probably would say, wait a minute, don't. You can't fool us. All it takes is 2 or 3 days. One of these cells divides. One cell makes 2. Factor 2 is not such a big deal. But it's important. But that's not the whole story, because if we look and try to figure out when are these cysts germinating, that's what this figure is trying to show you. So all this color in the panel, let's take A in the upper left, all of that color is the bottom temperature from 1999 to 2008, for that decade, the average bottom temperature. On the right is the temperature 10 years later. You can see the reds are warmer colors, right? And if you subtract one from the other, this one in C shows you the difference. It shows you how much warmer the bottom waters have gotten over that 20-year interval and where it's gotten warmer. And you can really see that this area, that's that 2 to 4 degrees Celsius there, that red color, that whole region over 20 years is warmed that much. And if we then look, this little green arrow, green circle on the right is how many, that's the cyst seed bed, and the one up in Barrow too. We can actually calculate when these cysts are going to germinate and how many. And you can see that 20 fewer days are needed for these cysts to germinate now compared to the prior decade. 20 days, if these cells divide every 3 or 4 days, you get division after division after division. It's no longer a factor of 2. It's a factor of 8 or 10, and you can start really building up a population because the growing season's longer, and they're just going to take advantage of it. So that was something about the cysts. We'll definitely come back to that. We also looked up in the water. So now we're looking, if you look over in the lower left here, we're up in the water at what we call the vegetative swimming cells here. And that's what these 3 panels that are colored on the right are showing. And I won't go into the details. There are 3 different cruises, one in 2018 at the top, 2 in 2019. And on the right are these diagrams that oceanographers use. They're called TS diagrams. They tell us a lot, but again, I won't go into the details other than to say, look at 2018. This bloom occurred right directly above that cyst seedbed, where all the cysts were so abundant. So, and then if we do look at this TS diagram, we see that those cells are all associated with this Alaskan coastal water, which is the water that hangs out much closer to the shore and is not typically associated with populations coming up from the south. I'll tell you more about that later. This bloom in 2019, we had 2 cruises in 19, is clearly just shot through Bering Strait. It came from the northern Bering Sea, established down there, and was just jetted through Bering Strait, and we caught it there near Kotzebue Sound. And you can see it's in different water. It's in this Bering summer water, which is the central current pathway right through the Bering Strait. And then up here later in that same year, we have a bloom that's off, that again is near the cyst seedbed. So what we've done is come up with what we call a conceptual model, and that's what you see here. So we're showing you 2 different mechanisms for blooms that occur up there. One of them is what we call these transported blooms. You saw a good example. These originate someplace in the Bering Sea, and they come up and they move into the Chukchi Sea. And you can see in the bottom here, the little diagram argues that these blooms are up there in the surface, they're growing, they're photosynthesizing, but when they make cysts, they fall down to the bottom, and that's occurring in that Chukchi Sea cyst bed area. And historically, the water's been really cold. What did I show you about the cysts? If it's really cold, they can't germinate. And so what you get is year after year of these transported blooms coming in, dropping cysts that don't germinate. What are you going to do? Big inputs, no losses. You keep building up the cyst population, and you get these huge distributions that we've mapped out. So we can explain why there are so many, and it's because these blooms from warm waters to the south are coming into cold waters, dropping cysts that can't cycle. But then you heard me say, for 2018, you said it was warm. And indeed, in 2018, 2019, that organism was able to do its entire cycle right there. And that's that second mechanism for blooms up there. One, transport them in from somewhere else. Second, let them germinate from the sediments. And it's like a double whammy, because some of these transported ones are still coming into those waters, but they're also having their own little cycle. And so that's conceptual model. We will come back to it. So then I want to talk about a cruise we just had, 2022. We were supposed to be on that beautiful big icebreaker. Because of COVID and lots of other things, they put us on this little boat called the Norseman. It's really not to scale here. That shows you the Norseman, and it was truly an adventure. But there was one advantage of that reassignment of vessels, and that's what you see here. Instead of giving us 1 month of time, they felt badly because we could have put 44 people, 44 scientists on the Healy. We could put 16 on the Norseman. So they say, okay, we'll give you 2 months instead of 1. So we actually were able to take that boat from Nome, out here in the Bering Strait, up, all the way up to the top, Barrow, and then all the way back down again, sampling the whole way. So there's one snapshot of what's going on in the water, snapshot number 2 coming down. A lot of people got off the boat, new crews got on, and then we went up and back again. Never have I ever had that kind of coverage of a small area trying to follow the bloom, and it was incredibly educational. And one of the things that we had that really made it a worthwhile cruise was a special instrument on there that you see on the left here. It's called the Imaging Flow Cytobot, IFCB. And what it is a submersible microscope that takes images of particles that contain chlorophyll, algae. And it does it incredibly fast. 8 to 10 images, high-resolution images, just like your phone, every second. Think of that, 1/1000. I just took 10 pictures of what was in the water. And that goes on 24/7, right, on this boat. And of course, we get hundreds of thousands of images, and humans can't deal with that. So how do you deal with it? You've all heard it, artificial intelligence, right? It's called machine learning. You train the software to identify these cells the same way you have the image, facial recognition in airports and so forth. It's the same exact idea, except we are processing hundreds of thousands of images on board, real time, as this instrument is sampling. So you see the intake coming into the ship, going through the IFCB, data is processed, and we know exactly what's in the water we're going through. Well, we left Nome, you see, coming down here on that first leg, and all the blue, each dot is an IFCB measurement. So that's basically every 20 minutes. And the little arrows that you see are vectors of the way the water's moving under the ship. It's from something called an ADCP. So we know the direction of the water, we know how many cells are in there of Alexandrium. You can see nothing, 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 nothing. You get over here to the western part of St. Lawrence Island, all of a sudden, really big numbers. These are 10, 20,000 cells per liter. In the southeast, if you get 1,000 cells per liter of Alexandrium, they'll close down everything. This is 20 times that already at the very beginning of our cruise. And notice, if you look at these vectors, it really looks like these cells are coming from the west, over there in the Anadyr. And I think I have, yeah, this is the Anadyr current as it moves up through Bering Strait, and that's the Alaskan coastal current. you can see the Alaskan coastal current basically has nothing in it. It's all blue dots, no Alexandrium. So they were coming from the west. And that becomes a big part of the story we're trying to solve. And notice you get up here to the middle of Bering Strait, a little diamede, and they're all right there in the middle of Bering Strait. The vectors are really long, the water's ripping through there, but there's clearly a transported bloom. We're capturing it beautifully with this IFCB. But these numbers are so high, we're realizing we've got to warn people. Never on a cruise I've ever been on have we got data real time where we have to say the people up there have to be worried about what they're eating because there's no monitoring going on. So we were issuing these advisories to all the communities in the north saying, be very careful what you eat because everyone up there is a subsistence harvester. They eat everything imaginable from the ocean. And you know, there's just so many ways things could be poisonous. So we were just telling them it's dangerous out there. And that was just with the first few days. And then it just kept continuing on and on. But here, at the very least, take that conceptual model, at least for that year, I'm going to cross off that pathway with the Alaskan coastal current. I don't think that wasn't functioning in 2022. These cells were coming from the west, so we had a little more knowledge. Okay, so remember that I said we mapped out and we added that same cruise and other cruises, this extension of that cis-bed, and we're wondering, well, why isn't that functioning? Why wasn't that showing up in the cruise? And one of the ways, well, I'll get to that in a second. What we think from everything I just said, these are those 4 snapshots I talked about. Leg 1, northbound, so going up to Barrow, coming back down, going up again, over here, and then coming back down. You can see the bloom that we picked up near St. Lawrence getting heavier and heavier and extremely heavy here. But part of the point down here is that we saw no evidence of that mechanism number 2. If there were cysts germinating and starting a bloom, they would have been doing it up here and up here, and we just don't see any cells there at all. So basically, we were seeing that vector bloom, but we didn't see that second mechanism. And you could wonder why. And these are all animations. It's back to this diagram. Remember, we'll have a quiz later. So here you saw the 85-degree days. Here were the temperatures, 2018, 2019. Where do you think they were in 2022? Way over there. 1 to 2 degrees. And you already know then the difference between 8 degrees and 1 degree. You know, it's going to be a huge difference, months, in the amount of time that it's going to take cysts to germinate. So basically, bottom temperatures were so low in 2022 that germination would have taken like 40 days as opposed to 10 days that we saw just a few years earlier. And that, I think, is a perfect explanation for why we weren't seeing that localized bloom. And you can see it even better here in this nice series of plots. So what we've done here, here's the little box where the cyst seed bed is. Over here are the temperature records from 2003 to 2022 for that little box. And you can see all the different temperature patterns, but notice 2018 and 2019 are the warmest in that whole record from 2003. Notice also, though, 2022 was the coldest. So if you use a little bit of math and make this plot here, it's kind of an interesting plot. This basically is the date at which you reach that 85-degree day. So if you're a cyst on the bottom, and it's 2003, and it's cold, you can actually calculate how long until you get 85-degree days. And that's what's plotted here. You can see that in 2003, it would have been somewhere near August 2nd. But if you just look at the general pattern, you can see how cold it was all these years. The cysts aren't germinating until August 7th, August 12th, August 17th. Notice these are the years we started our program, 2018, 2019, when we saw the really warm water. So we were lucky, in a sense, to have seen really warm conditions in what has been colder waters. So most of the time, that seedbed is not functioning. It's just accumulating cysts. But it's every few years that it does start functioning. So in one of our papers about this, I called it the sleeping giant. That it's just waiting, and suddenly it wakes up and it's big and it's nasty. And that's what you're seeing here. So what you're seeing is this significant interannual variability in the bottom temperatures, nearly a month difference in the onset of cyst germination between 19 and 22, and multiple years when it's cold are going to help you build up that cyst and seedbed. Okay, so let's go back to here. Let's figure out where these cells, or try to figure out where these cells coming from. So you've seen the data before. This all suggests something's coming from the west in this Gulf of Anadyr. So we hypothesized that there's another cyst bed out there in Russian waters that we can't sample somewhere in that region. We don't know where. But let's have a little experiment where we basically hypothetically put a cyst bed here. Just it's someplace in the Gulf of Anadyr. We put another one right in that part of the eastern Bering Strait where we measured a lot of cysts. And then we just used a numerical model of the way the ocean moves, and what, not just theoretically, but the way the ocean actually moved, in this case, in 2022. And you can see that these little particles would have traveled here. Notice, to get to St. Lawrence, what do? Some of them are going south of St. Lawrence. That's exactly what, see, we saw here, some of the cells are heading south of St. Lawrence, some are going that way. That's exactly what is happening here in this blue. And then they head up through Bering Strait. But notice this red cyst accumulation also is a viable pathway. That Alaskan coastal current should have been able to bring cysts and cells up a little slower than into Bering Strait, but still, they get there. They get there eventually. So both of those pathways are valid for transport, but we still think the biggest and the most important one is the one to the west. But we have a problem. This is back to our 85-degree day. This is the same plot you saw, this upper left is the same plot you saw earlier. It's just squashed down because I had to make the scale the same as the one below, which is for this little hypothetical spot in the Gulf of Anadyr. And what it shows you is that it's much colder there. And those cysts in that hypothetical spot aren't going to germinate until the end of August, mid-September, even October or November. They're certainly not in time to get to the July or August when we saw them in Bering Strait. So we've got a real problem. We're saying there's a cyst bed, but the water's too darn cold for the cysts to be germinating and getting to us. So all we can really say is that there's probably something else. There's probably a shallow water area, a bay or something that is a source. We've got colleagues in Russia we're trying to work with, but as you can imagine, the Ukraine conflict is making that really difficult. So we have a mystery. Perhaps there's another cyst bed over there, and we're going to try to find it at some point. Okay, a couple more things here. Why did that bloom end? I showed you that conceptual model. The bloom goes along, and at some point all these cysts fall. Did we see that? Well, here you see some other way you can use the IFCB data. It gives you not only images that you can identify, but you can measure the volume. Here's an Alexandrium cell that's one of those little gametes that fuse together. And here's one that's called a zygote. That's the precursor to the cyst. And just over these 2 days, I've only showed you part of the record, you can see that the volume of the Alexandrium cells increased dramatically. And we've got a lot more data to show you, but basically there's no doubt in our mind that we captured the end of the bloom where the cells went into what's called sexual reproduction, making gametes that fuse to make those zygotes that then fall down as cysts. And so if you go back to try to look and count them. Here's a little transect. We had 2 cruises during that year. The summer, pre-bloom, The cysts in the bottom sediments along that transect are this orange. And you go from near shore on the right to offshore on the left, and you can see the abundances in the summer, before the bloom. After the bloom, you can see how many new cysts were deposited. Sometimes factor 6 or 10 more at the same location. So we saw a huge deposition of cysts. We know when it happened as well. And so if you go back to this, what did we observe in that cruise, or those cruises? Transported blooms? Absolutely. You all saw it. They were there. No question about it. We saw this deposition of cysts into the cold waters, where they would historically not germinate and build up that huge cyst bed. What we didn't see was that locally initialized bloom. We've seen it other years, but we didn't capture it in 22, and we know why, because it was really quite cold. So, final bit of the story. I said that this organism makes this neurotoxin called saxitoxin. It's actually a family of toxins. There's more than 24 of them. And you see they have these little names. This is saxitoxin, GTX 1 and 4. And this is just a table of their potency. When they affect us, they bind to this thing we have called a sodium channel. And it basically is the way that we flex our muscles, and sodium moves in and out of our cells. The more potent that toxin is, the more it holds on to that. And Vera did this work for her thesis and so forth. The more potent that particular compound is, the longer it holds on to that sodium channel spot. If it's weak, it's on, it's off, it's on, it's off, and it's less toxic. So you see here, saxitoxin is sort of the standard, high potency, 100% potency. At the other extreme, these C toxins down here, they're from anywhere from 1/100th to 1/1000 as toxic, because they don't adhere to that same sodium channel well. Well, why am I showing this? Because these little pie diagrams show you which toxins were in these bloom cells throughout the bloom at these different points. We're here, 1, 4, 5. You can see where they're taken. And the first message is those are all pretty much the same. It's like a fingerprint, a toxin fingerprint that tells us that population is carrying toxin A, B, C, D, and it's the same throughout that 2-month period, essentially. Point 2, though, is that if you look at the colors, most of it's GTX 1 and 4, neosaxitoxin, and some GTX 2 and 3. So the 4 most potent toxins are the 4 that were in that bloom. It couldn't have been, essentially couldn't have been more toxic. And there were huge numbers of cells. So that's an eye-opener as well. But we also try to use these toxin fingerprints to help us figure out where these blooms come from, connectivity. So I'm back to where does this bloom come from? Maybe we can find out from the fingerprints, but they're plotted 2 different ways. On the left, you see the pie diagram, but it's the same thing as on the right, where you see the histogram of all of these isolates we've made of this species from the Bering Sea, Bering Strait, Chukchi Sea, Chukchi Shelf, and Barrow Canyon. And it's hard, but basically if you looked across there and tried to find a bar where most, 50% of it, is this teal blue, and maybe 25% of it's this red, and a little bit is yellow. Go across here. You know, this, you don't see it until you get to here, way at the end in Barrow Canyon. You're almost, in the Hudson Bay. You're way east at the very end of this pathway. It makes no sense that this is our origin. This is the deposition of some of our cells, and it increases our mystery. We have not been able to identify a source region from all the things we took. Of course, we're working in American waters, right? From the Bering Sea all the way up. Now, I did this one you understand. Once again, we have to have another region somewhere. I did a study years ago with colleagues well known to my friends here. This is with Tatiana Orlova from Vladivostok, where we got samples from Kamchatka and all along that Russian coast. And this is yet another kind of way of showing the toxins. It's similar to the pie chart there. The colors are the same. And you can see, again, do you see any isolate anywhere that looks like this? I mean, this one's all dark blue, this one's all black. You know, maybe here there's a little bit of this teal color. But even in these samples we have from that part of the Russian coast, we don't have a match. What's the one area that's missing? It's in here, this little Gulf of Anadyr. So I still have hopes that our origin's there, but we don't know. But we're trying. Okay, so what does this all mean to the people up there? I mean, this is the community of Diomede, right on that tiny little island in the middle of Bering Strait. This is Little Diomede, right next to it is Big Diomede, which is Russian. It's only, I don't know, it's a few miles away. 50, 60 people live there. They get all their food from the ocean. Here's another one. This is Gamble on the west part of St. Lawrence Island, right where we were seeing that bloom. And these people eat virtually everything from the ocean that you can imagine. And historically, all over the world, for PSP, we've worried about shellfish and crabs, and occasionally some forage fish, you know, like herring or something, being vectors for the PSP toxins. But up in this region, all of these walrus, seals, whales, sea stars, tunicates, everything that they eat are potential vectors. And so there's still a big risk. Now, one thing people are fortunate about, they don't eat very many clams up in that part of the Arctic. They're too deep offshore, they don't eat a lot of them. If they did, here's some data from a colleague, Kathy Lefebvre, during that 2022 cruise, showing these red and orange little scallops are indicative of clams all along that cruise track that were dangerous to eat above the FDA's regulatory limits. And so these offshore shellfish, dangerous for sure, but the people don't tend to eat them. But guess what does eat them? Walrus. Walrus loves eating them. But in addition, I found this was great to see. This was a Facebook post that someone sent to me. One of the things they love is when there's a huge storm, all these clams wash up on the beach and they go and collect them. And that's when they eat shellfish in huge abundance. And here's somebody with bucket after bucket after bucket of clams about to bring them to the whole community to eat. And when this happened, it was during our cruise, and we had to try to figure out who this was, send messages to that community to not eat those clams, okay? The storm's blessing, so many clams, they were so thrilled. And then here come the scientists saying, you're not gonna eat that, are you? And so, anyway, that's one big risk. There's another risk, which is that we don't know the toxin levels in these different animals, like seabirds and so forth. So what part of a seabird can you eat? What part of a seal can you eat? Where is the toxin? We're learning. We have all these studies ongoing to try to learn that, but we're still just beginning. And more importantly, perhaps, we don't know the lethal doses for a lot of these animals. We know seabirds die very quickly from this toxin, but we're still learning what's the lethal dose for a walrus, for a whale. And that brings up food security issues. So it may be that the bigger issue isn't that people are going to get sick, but there's going to be a lot of mortalities of a lot of the things that they do eat. But we'll just have to see. And the final thing I just wanted to point out, this all may seem esoteric, but one of the things I've really been trying to do is to find local people who had knowledge that goes back hundreds of years of the history for these regions. Are there times of the year when you eat shellfish and other times when you don't, or that kind of thing, like we have on the East Coast. And someone sent this to me. It's a National Park Service publication, unfortunately with no dates in it. But at the bottom there, it's talking about this spot out here in Seward Peninsula, this little red spot. This Ipnarak was the location of a red tide at one time, which caused many deaths. So at some point, a lot of people died in that area. And notice this is our bloom from 2022, and that area is right there. So we had this huge number of cells right there. And of course, we're telling all the communities, be careful what you eat. But in some year past, many, many people died there. And we don't know when it happened. We don't know what the vector was. We do know that there's a lot of fishing that goes on there is used as a site for fishing herring. Herring is a filter feeder, can definitely accumulate this toxin as it swims through blooms. Okay, so in any case, I'm through. I just want to summarize. So we've shown that we've got at least three of these huge cyst seed beds up in the Alaskan Arctic. We've seen these high concentration of transported cells every year since 2018. So I don't know, we weren't there before. The fact that it's been every year makes me wonder if it's getting more frequent. Don't know. It's warmer, it may well be doing that. Some blooms have been extraordinarily dense, like the one in 2022. And we know that in some years, but certainly not all years, bottom waters are warm enough to have that mechanism number 2 for these cells to be able to function, the sleeping giant cyst beds to function. Now, I'm one who believes that it's going to continue to warm up there, and that we're going to have more years that are going to be warm like we saw in 2019 and 18, but we'll see. We also, in terms of, we don't know where these cells are coming from. There's a hydrographic pathway from the Gulf of Anadyr that could do it. There's one from the south as well, from the eastern Bering Strait. We still don't know. So what you've seen, though, I was talking with my colleague Mark Wells. He wrote a paper about HABs and climate change. And one of the things we say in that paper is there's a potential for our HAB species to get worse because of warming climate. And this one may be a winner, this one may be a loser. This is one of the only examples we have, a very clear example of a HAB expansion due to global warming. Because we've seen in 2018, 2019, the water's warm enough for this species that's been trying to colonize this area to finally get a condition that allows it to bloom. So it represents a major threat to the subsistence communities that rely on such a diverse array of marine resources for foods, and many of them can accumulate these toxins. And so with that, I think this is sort of a fitting, it's the wrong season, but it still sort of shows you, to me, the challenge and the mystery that these people are facing as they try to live the way they have for tens of thousands of years, where this new threat is coming. So thank you for your attention. Happy to take questions. Sure, yes. Yes, you, yes,
I'm
sorry.
In samples that you took, what was the percentage of Alexandria on all the algae?
Yeah, okay. So that's what's great about the imaging flow cytobot. Actually, if I were a little more adept right now, I could actually go onto a data portal and show you every single IFCB image, and you could see it visually. But the answer is that during the beginning of the bloom, the cells were maybe 10–20% of what was there in terms of all the other species. When the bloom really, the colors turned red on the maps, when we had 150,000 cells per liter as Alexandrium, it was like 80%. Not quite what we would call a unialgal bloom with single species, but very close, which one of my students is trying to help figure out what's going on. It's one thing this is not a fast grower, so there's a lot of things that have to happen for a species to dominate. And it can happen because they grow really fast, or it can happen because they have behavior where they swim and the currents congregate them in a certain place, but it also can happen if they release chemicals that are not toxins, but we call them allelopathic chemicals that inhibit other things trying to grow around them. And this species is well known to do that. So maybe that's why it got up to being almost a monospecific bloom. Sure, yes.
You touched briefly on levels in different animals, and are you starting to see in the migratory patterns if that's going to carry it someplace else possibly?
All right, well, there's several questions there. First of all, when we are definitely getting levels in the animals and starting to have answers when someone says, can I eat my seal? And we've been asked that. Well, now the answer is, well, we're quite sure that there's no toxin in the blubber, in the muscle, but it is in the kidneys, it's in the liver, it's in the stomach potentially. And so there's certain things that you shouldn't eat and some that you could. Now, you're talking about migration, but are you talking about the movement of the algae itself or the animals themselves?
Do they retain that toxin?
The toxin, yes, they will retain them, and they can live with low levels of the toxin in them, but they're not going to, eventually that toxin is going to be excreted, and it's going to be broken down, so it's not something, we don't worry a lot about dissolved toxins in the water. It's the particulate toxin made by the cells. So yes, the toxin can move with these animals, but that's not really a concern to us.
Hey, Don. We have a question, a hand raised on Zoom whenever you have a moment to take it.
Okay, great. I can't see the hand from here. So.
Yeah, no problem. Bernard has his hand raised, so Bernard can unmute and talk. And Don, if you wouldn't mind, stop your screen sharing, and then we could see you a little bit bigger on Zoom. Thank you. Perfect.
Hey, Don, I really want to thank you for giving such a wonderful talk. It's fantastic that you were able to do that, and thank you, Vera, for setting it up. I've got a probably naive question from a kind of a forest ecologist, but it seems like we hear a lot about king crab fisheries, fluctuating wildly, and do you see any possible connection to that important economic driver?
Well, the connection I see is not one that's direct. It's more that the forces that are acting on the king crab, and I don't know exactly, but I'm quite sure it would have a lot to do with bottom temperatures. And that they, I've heard you lost on it was a king crab or the different crab that it was too warm or too cold and they all died and the whole fishery was wiped out for a year or so. That, the king crab's problems I think are related similarly to bottom temperature, and the only real connection to what I'm showing you is that, yes, indeed, water temperatures are warming and their bottom water temperatures are warming, and just like the king crab may be affected negatively, Alexandrium is being affected positively, as are many other organisms. And I mean, I've heard they're now catching pollock up in the Chukchi Sea and so forth. A lot of animals are moving up there, and unfortunately among them are these toxic algae. Yes, please.
Do the cysts die if what they're in is cooked, heated up?
Okay, well, I'm going to answer a question you didn't ask, and then I'll come back to that. Because a common question that I wish I had thought to preempt.
Oh, and can you repeat the question
Okay.
I was supposed to ask you?
All right, so the two questions, one of them was, do the cysts die? I'm going to stop it there. And then the second part of it is, do they die if you cook them? But the first one is that cysts can live for decades and even 100 years in the sediment. They have built a thick wall around them. Their physiology is such they can be buried deep in sediment with no oxygen, and their metabolism is still able to keep them dormant, and even 100 years later, you can germinate them. So that's why I say that cyst bed can grow over the years as you drop all these cysts, and it's cold, but eventually it's going to warm up, and then they can germinate. But the other part of your question is, can you cook things so that the toxin gets destroyed? And unfortunately, it's not just this toxin. Virtually every toxin we all study does not get destroyed when you boil it or heat it. The best way you protect people is just to keep them from eating them. But cooking, and in fact, there's an argument that if you cook a certain way, like with vinegar, some acid, you can actually convert some of those really weak toxins on that table I showed you back into the really potent ones, just because of the acidity of what you're cooking them in. So, but you don't destroy it. See, there's another question over here. Sure, yes.
Can you see the blooms on satellite?
Well, these blooms, almost never, okay? Even at the densities we saw, we had a friend, you know, in Brazil actually, who was looking at images the whole time, a lot of clouds, difficult to see, but when there was a clear day, you could see some chlorophyll in the water, but you could not see it had any of the features of what we were mapping out, even when it was 80% Alexandrium. It takes, we were seeing like nearly 200,000 cells in a liter of water, like in a quart. It takes at least 1 million, maybe 2 million in that same volume for us to see red water and a red tide. And so even at a dangerous level like this organism is not visible from space. Now that being said, there are other HAB species. We've got a colleague here from Florida where they have this organism that reaches 30 million cells per liter, and it is visible from space, and satellites are very useful in monitoring that. Out here, we have not had any good success with satellites.
All right, I think we'll take one more. Courtney, are there any more online?
No, there are no more online. Thanks though.
Well, maybe I'll ask one
Well,
last.
he's got one.
Okay, we've got one, okay.
I just, does the toxicity of Alexandria vary over its life cycle? Is it just simply a matter of mass during the bloom?
Okay, first of all, it does vary a little bit over the life cycle. If you think about it, 2 cells fuse to make a zygote, that then becomes a cyst. So a cyst can be a couple times more toxic than the other cells that produced it. That's not a big deal. But if we grow these in culture, and you know, if you just take a flask and dump in some of the nitrogen and phosphorus and grow them, they do something that's called batch culture growth, where they sort of lag for a little bit, and then they grow and they divide and divide and divide and divide, and eventually they plateau. And during that exponential phase, as we call it, they keep their toxicity quite constant. But if we really starve them and they get to this plateau phase and we extend it for a long time and keep them alive, or we use other culture methods to do that, then they can actually start switching that fingerprint of toxins. And that can change their potency. Remember, I'm saying some are really potent, some aren't. And so at certain stages of starvation, you produce more of this congener, we call it, than this one, and that can affect your toxicity. So you can actually get, by manipulating that growth, we can get cells 10 times more toxic than others grown with a slightly different medium. But we think that mostly is happening in the lab, where we're, you know, it's sort of like putting something on a frying pan and measuring its metabolism. It's running around as fast. It's not normal. You know? So typically, we think it's quite constant during the bloom.
Okay, thank you so much, Don, again. Thanks to you who joined online. Thanks to all of you for coming. The next evening talk will be on Thursday, March 21st at 7. And Jerry Borchert from the Health Department will be talking about keeping our clams and crabs safe here in Washington. So please join us then. Please take some muffins with you on your way out. We have so many muffins.
That's the day I leave.
And we really appreciate you